Okay, go.
I'm asking you guys questions.
No.
Okay, here's your stem cell. How many chromosomes do you see there?
46.
How many chromosomes do you see here?
23.
3? 23 times 3.
Oh, it's 2. 1, 2, 3, 4, 3.
Really?
No.
6?
That's not a good one.
That's not a good marker for us that are making the video.
6.
Okay, do you see
6
6?
chromosomes. Yes.
Are you sure you don't see like other funny numbers?
No.
6 chromosomes.
Remember, this is what's taking place in here is that each of these chromosomes start off as one single or double-stranded DNA, and then it goes through DNA replication to become the 2 strands, or the two chromatids. Okay, this is all what we kind of went through before when we talked about mitosis. So in interphase DNA replication, you take your double-stranded DNA, separate them, make semi-conservative, make a new strand for each one, so you get a replicated chromosome. So what you see here are 6 chromosomes that are replicated. Okay, so 6 in this case, since it's a stem cell, is our diploid number. Okay, that diploid number now, we want to make gametes, so we're going through meiosis. And by going through meiosis, the first stage of meiosis is going to be to separate our pairs, our homologous chromosomes, into our new cells, our haploid cells. So essentially what you're doing is you're going to take each of these and separate them. Now how many chromosomes are in each of these cells?
3.
Which is half the number that we started with, right? So each of these formed cells has half the number of chromosomes that we started with. But notice, they're still replicated chromosomes. We started with 6, now we have 2 cells that each have 3, so these are haploid cells. But they're still replicated, they still have sister chromatids. So going into meiosis 2, each of these is now going to split again, but what's going to happen is that in meiosis 2 we're going to separate the chromatids. So we have something like this. We have 4 cells that each have how many chromosomes now?
3.
Each one has 3, the haploid number. So all we've done in meiosis 2 is we separated the chromatids and made them their own separate daughter chromosomes. And that's the main, of course, you go through each individual stage, but when we talk about meiosis 1 separates your homologous pairs, separates the pairs into forming haploid cells. But we have to continue because these haploid cells have to break down the chromatids. So to complete meiosis 2, we separate the chromatids to end up with 4 cells that are all still haploid. Half of the number of the original 2 sets. These, if they're sperm, will then swim over and fertilize the egg, and then these 23 would fertilize the other 23, and you get 46 again. Or in this case, these 3 would fertilize another 3, and you get 6 again. Okay, and so now as we're relating this to our body, yeah, we have 46 in our typical cell. Those 46 go through meiosis 1, then we have 2 cells that each have 23. Those 2 cells, those 23 chromosomes in each of these cells still have sister chromatids. So in meiosis 2, we take these 23 and separate the chromatids into 2 separate cells that each also have 23. That should be the easy part. The hard part comes when you're looking closer at what's happening here, because These are homologous, meaning that they're similar. They're coding for the same general information. But you're going to exchange information between these homologous pairs. And by exchanging that information, it leads to greater genetic diversity over here. So that means that if these are sperm, not one of these are going to be the same. We're talking about trillions of different possibilities that are occurring. Okay, and that's the basis of what you want to be gathering from this. Yes, you need to go through each step. We talked about what's happening in each step of meiosis. Okay, but this is the overall concept. Questions?